In this video, we are going to learn what is respiration in plants. Respiration in plants is basically a cellular respiration in which oxidation and reduction reaction takes place simultaneously. It is basically a stepwise breakdown of food which involves oxygen and releases trapped energy. At the same time, we call it metabolic process. It is basically a catabolic reaction in which energy releases. Now we are going to learn what is respiratory substrate. Respiratory substrate is basically every compound of carbon which upon oxidation releases energy. Mostly respiratory substrate is glucose, while other respiratory substrates are fats, proteins, organic acids. Now, what is not a respiratory substrate? It must be a water, vitamins, minerals and hormones. If someone asks from you, does plant breathe or not, then simultaneously we can say, no, plant doesn't breathe, but there is a gaseous exchange which takes place in the presence of stomata and lenticels. Basically, maximum parts of the plants complete their oxygen requirement not by large diffusion. Energy need in plants is always less than that of animals and other higher or complex animals. What is the process and site of respiration? The process of respiration involves several steps, one of which is glycolysis, which takes place in cytoplasm. Second one is oxidative decarboxylation, that is link reaction, and takes place in the matrix of mitochondria. And third one is the Krebs cycle, which is also takes place in the matrix of mitochondria. Now we are going to learn what is glycolysis. As in other animals, we can say glycolysis is the process in which glucose is broken down into pyruvate. But in plants, as we know, reserved carbohydrates are sucrose and starch. So, the glycolysis takes place not only in cytoplasm but also in plastid, because we know that reserved carbohydrate, that is starch, is stored in plastids, right? Here is the plant cell in which glycolysis takes place in cytosol as well as in chloroplast or plastid. Now we are going to start the process of respiration. In the process of respiration, two basic phases are involved. One is energy investment phase and other is energy yielding phase. For energy investment phase, this process takes place in the cytosol. In other animals, we can say glucose is broken down into two molecules of pyruvate, but in plants, sucrose is immediately broken down into glucose and fructose by using enzyme invertase. While sucrose is also broken down into UDP glucose by using UDP and sucrose synthetase enzyme. Now the glucose is immediately broken down into glucose 6-phosphate by utilizing energy of ATP and enzyme hexokinase. And fructose is immediately broken down into fructose 6-phosphate by utilizing energy of ATP and enzyme hexokinase. Both glucose and fructose are interconvertible using enzyme hexose phosphate isomerase. Similarly, UDP glucose on the other hand is broken down into glucose 1-phosphate using energy of UDP and enzyme UDP glucose phosphorylase, which is immediately converted into glucose 6-phosphate by a reversible reaction using enzyme phosphoglucomutase. Glucose 6-phosphate is again interconvertible into fructose 6-phosphate. Now, fructose 6-phosphate is converted into its isomer using enzyme phosphofructokinase into fructose 1,6-bisphosphate and utilizes the energy of ATP. At this step, fructose 1,6-bisphosphate is immediately converted into two molecules. One is G3P, that is glyceraldehyde 3-phosphate, and other is dihydroxyacetone phosphate. Normally we can call it DAP using enzyme aldolase. Both G3P and DAP 3 carbon compound that are immediately interconvertible using enzyme isomerase. All this processes takes place in the cytosol, but for plastids, starch is immediately converted into glucose 1-phosphate and glucose using enzyme starch phosphorylase and amylase. Both glucose 1-phosphate and glucose is immediately converted into glucose 6-phosphate by utilizing the energy of ATP and enzyme mutase and kinase. Glucose 6-phosphate moves from amyloplast into cytosol, and all the process of conversion of glucose 6-phosphate to fructose 6-phosphate and fructose 6-phosphate to fructose 1,6-bisphosphate takes place in the cytosol. Sometimes glucose 6-phosphate, that is 6-carbon compound, is immediately broken down into its triose phosphate, which is again then move toward cytosol from chloroplast. Till that, glucose 6-phosphate, fructose 6-phosphate that are interconvertible are hexose both in cytosol and in plastid, while G3P dihydroxyacetone phosphate, which are also interconvertible, and triose phosphate generated in the plastids are also interconvertible, and these are all 3 carbon compounds, so we call them triose phosphatases. First of all, we have seen that sucrose is immediately broken down into 2 molecules like glucose and fructose, and on the other hand, sucrose is immediately broken down into UDP glucose in the presence of enzyme sucrose synthetase. Now, sucrose is immediately broken down into glucose 6-phosphate, only addition of phosphate group at 6 carbon. Then fructose 6-phosphate, glucose is immediately converted into its isomer, that is fructose, 6 carbon ring to 5 carbon ring. Fructose 6-phosphate utilizes the energy of ATP and again attaches another phosphate group on 1 carbon and changes into fructose 1,6-bisphosphate, and fructose 1,6-bisphosphate changes into 2 molecules, that is glyceraldehyde 3-phosphate, we call them G3P, and DAP, dihydroxyacetone phosphate. Both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are interconvertible using enzyme isomerase. From that, energy yielding phase starts. Fructose 1,6-bisphosphate is converted into G3P and DAP. G3P is a 3-carbon compound and both G3P and DAP are interconvertible. The difference between these two groups is that there is a presence of aldehyde group in G3P and ketonic group in DAP, that is dihydroxyacetone phosphate. G3P is converted into 1,3-bisphosphoglycerate. From that, we use only G3P because dihydroxyacetone phosphate is also converted into G3P, and we again start the whole process using G3P. Like G3P is converted into 1,3-bisphosphoglycerate, and we have 2 molecules of 1,3-bisphosphoglycerate, one from G3P and other from dihydroxyacetone phosphate. G3P is converted into 1,3-bisphosphoglycerate, and it is the first step where oxidation takes place and NAD positive is converted into NADH. 1,3-bisphosphoglycerate is immediately converted into 3-phosphoglycerate and produce 1 ATP molecule using enzyme kinase. At this step, only 1 ATP is not produced, as we have already discussed. G3P and DAP are interconvertible. So we have 2 molecules of ATP and 2 molecules of NADH produced. 3-phosphoglycerate is immediately converted into its isomer, that is 2-phosphoglycerate using enzyme mutase, and 2-phosphoglycerate is immediately converted into phosphoenol pyruvate using the enzyme enolase and causes the removal of 1 water molecule. Phosphoenol pyruvate at the end causes the formation of ATP from ADP and formation of pyruvate molecule uses enzyme kinase. Now, one most important thing we have to consider is all the ATP conversion into ADP and all the ADP conversion into ATP involves enzyme kinase, and its starting depend upon the substrate. Like first of all, we have seen that glucose is converted into glucose 6-phosphate using enzyme. Hexo is basically a substrate and kinase is an enzyme which convert ATP into ADP. At the end, phosphoenol pyruvate takes an alternative path and uses enzyme PEP carboxylase and converted into oxaloacetate. This oxaloacetate in cytosol is converted into malate using malate dehydrogenase, and this malate again move towards the matrix of mitochondria and used in Krebs cycle. Till that, we have learned the process of glycolysis in cytosol as well as in plastid. So, in the process of glycolysis, 4 steps are irreversible. First, 3, 7 and 10. These are rate determining steps, but out of all these steps, only 3rd step we call them rate determining step that uses enzyme phosphofructokinase. 2 molecules of pyruvate produced at the end of glycolysis is immediately moved toward mitochondria in the presence of oxygen that causes the aerobic respiration, and if oxygen is not present, these 2 molecules of pyruvate undergoes fermentation. Fermentations are basically of 2 types. One is alcoholic and other is lactic acid. Lactic acid is a 3-carbon compound which immediately converted into lactate and converts NADH into NAD positive using enzyme dehydrogenase. Alcoholic respiration reaction or alcoholic fermentation causes the conversion of pyruvate 3-carbon compound into 2-carbon compound acetaldehyde by the release of carbon dioxide and then converted into ethanol and causes the formation of NAD positive from NADH using enzyme dehydrogenase. Here is the list of structures in which 1,3-bis-phosphoglycerate immediately break down into 3-phosphoglycerate using enzyme kinase and causes the formation of one ATP molecule, and then 2-phosphoglycerate only its position changes from 3-carbon to 2-carbon and then it changes into phosphoenol pyruvate. By enzyme enolase, water molecule releases from this side, only CO remains. Then at the end pyruvate forms which is converted into lactate or acetaldehyde or ethanol in the absence of oxygen and immediately moves toward mitochondrial matrix in the presence of oxygen. In the next video, we will learn what is Krebs cycle or oxidative decarboxylation and link reaction.